Hello, and welcome to this special installment of the Future Grind podcast. I'm your host, Ryan O'Shea. As you know, this podcast is focused on exploring the ethics and implications of emerging science and technology. But this episode is going to be a bit different. I recently partnered with Lifespan.io to create the Rejuvenation Roundup Podcast, an audio version of their popular monthly article series that provides updates on the current state of life extension science. I've decided to share the first episode of this podcast with you here. It features some very noteworthy guests, including Dr. Alex Zavoronkov from In Silico Medicine and Daria Kulturina of the International Longevity Alliance. We hope that you'll subscribe to the Rejuvenation Roundup podcast and help us spread this important information. Now, here's the unabridged first episode. Hello, and welcome to the first episode of the Rejuvenation Roundup Podcast, where we explore the latest in life extension and anti-aging science with a dive into a month's worth of insights and new breakthroughs. This podcast is an effort of the Life Extension Advocacy Foundation. I'm Ryan O'Shea, and I'll be your host. Many of you may know me from Future Grind, a podcast that explores the ethics and implications of emerging science and technology. If you haven't heard Future Grind and want to check it out, I recently interviewed Leaf Vice President Oliver Medvedik about the science of life extension. You can find that episode and many more at futuregrind.org. But this is August, and that means that it's time to recap July 2018 in this debut episode of the Rejuvenation Roundup podcast. To find out more about any of these topics, you can visit lifespan.io/roundup. First up, July featured the first ever Lifespan.io conference in New York City. With 160 attendees and some great talks from speakers such as Aubrey de Grey, we're extremely proud of how the conference turned out. You can visit our website to see a conference summary, check out some photos, and view the video of Leaf President Keith Comito's opening speech. More videos will be released soon, and they will first be made available to our Lifespan heroes, without whom this event would not have been possible. Lifespan heroes support our work with monthly contributions, which enable us to fund more research. If you'd like to become a Lifespan hero, you can join online at lifespan.io. Thank you to everyone who has helped make our first conference possible. Our team is already hard at work planning a second edition for 2019. And now for some research updates. At the recent International Cell Senescence Association conference, held in July in Montreal, Oisin Biotechnologies presented promising preliminary data on mice survival after undergoing treatment for senescent cells. To find out more about this study and what it means, we spoke with Oisin Biotechnologies CEO, Matthew Schultz.
I'm sure this audience is familiar with senescent cells in general, these zombie-like cells that are damaged in such a way that their cell cycle is permanently arrested. And generally, these cells would be killed off by the immune system if they are created during injury. But as we age, we accumulate populations of these senescent cells that don't die. And so they basically sit there indefinitely, secreting inflammatory cytokines and generally poisoning their environment. And it's thought that this secretory phenotype is actually responsible for a considerable amount of the pathologies we associate with age. Oisin, like several other companies in this field, I think was really inspired by work done at the Mayo Clinic, where they had genetically engineered mice to let them kill these senescent cells with the drug. And the results were really quite remarkable, and some of the photos were published quite widely. You saw this decrepit-looking, emaciated-looking mouse and a plump, healthy-looking mouse, and they're litter mates. They're the same diet, same lifestyle, identical genetics. The only difference was that they had cleared the senescent cells with a drug from one and not the other. And this was really, really exciting, but the study was done in a way that was utterly impractical for human use. It required you to genetically modify the organism before it was born, effectively, so you could do this when it was alive. And so this really got us thinking about how could you do that in a way that was viable clinically, that could actually be a therapeutic. And it led us to develop this platform where we effectively deliver genetic programs, programs written in DNA that can kill a cell based on the genes it's expressing. We often say we're killing cells based on what they're thinking, not what they look like. And in the kind of an example with what was done at the Mayo Clinic, they were killing cells that expressed P16, and they genetically modified them so they could do that with a drug. And so in our first senescence studies or senolytic treatments, we actually started killing P16 expressing cells as well. But we were able to do this in normal lab mice without any actual genetic modification. So you just inject them with the genetic payload inside this exotic lipid nanoparticle that can get the DNA into the cells. And the cells that we want to kill get killed, and the other cells are unharmed. And the way we do this is by encoding something called caspase-9 with the DNA that we deliver. And caspase-9 is an apoptotic protein. It's part of the body's own cellular death pathway. So it basically tells the cell to shut things down and effectively recycle itself. So it doesn't cause inflammation like if you killed the cell with a chemical or damaged it physically. It just shuts down cleanly. And so we're able to use a combination of different promoters and repressors to selectively cause cells to commit apoptosis on their own. And we think this is super important because if you think of a senescent cell or a population of them in a human as you age, they're not like in a real tight ball where you can just cut it out like you might think of a tumor. They're distributed across tissues. And so a senescent cell is likely surrounded by lots of healthy cells. And if you deliver toxins or poisons to try to kill the senescent cell, you're invariably going to cause damage to the healthy cells. And so in our case, we don't deliver any toxin, any poison. It's just DNA. And the DNA might go into all the cells, but it will only activate in the senescent cell. And so a few years back, we decided to test this out. And so we got some naturally aged mice, and they were, I believe, 80 years old when we started, just normal lab mice, B6 lab mice. And we gave them doses of this. And we actually had it set up so that the DNA made the senescent cells express luciferase, like a firefly protein that we could see, and the suicide gene, the inducible caspase-9. And we were able to show that we could, in a dose-dependent manner, clear senescent cells from lots of major organs, the kidney, spleen, lung, seminal vesicles, fat. We had a whole bunch. We looked at it. And we were quite pleased with this in that it was able to recapitulate things that had been seen in the transgenics, but in a way that was viable. And so recently, and this is the data that we just presented, we wanted to see if it would actually make an impact on their overall lifespan. And so we had been developing different versions of these. The P16, like I said, we started with, which is the same general target that the Mayo Clinic guys used. But we also started doing work with another target, a P53. And we were using this a lot in cancer, actually, because a P53's kind of nickname is the guardian of the genome. It's capable of killing, it basically arrests growth of tumor cells, either stopping them from dividing or outright killing them. There's considerable overlap, though, between senescence and cancer from a genetic perspective. And so we found that certain cancer cells can be killed with P16 and certain senescent cells express P53. And so we decided that in this study, we would test both P16, P53, and a combination of the two. And so this is a larger study. We have about 40 mice, roughly 10 in each group. And we let them get relatively old before we started. So they're 106 weeks, so 2 years old basically, and it's pretty old for a mouse. So to put it in context, if you look at the survival graphs from large mouse labs, they'll basically show that by 2.5 years old, 50% of the mice will be dead, and by 3 years, they're effectively all dead. And so this is kind of the background data we're operating under. And we showed that if we treated these mice, we had considerable benefits. And I guess initially, actually, we were showing that we're seeing improvement in bone density, which was not a total surprise, but still interesting to see as we administer it in old age. I guess the critical data here, we had one control group that just got injected with PBS, and so they get injected once a month. And we're out now at about 2.5 years old. So the studies were in progress still, 6 months into it, or 6 months into the treatment phase, so 2.5 years since the start. And we have 60% of the PBS, kind of sham-treated mice are dead right now. And we found that if we gave the mice the the treatment that removed P16 cells, we roughly cut the mortality in half. So those have only 30% of them are dead. And when we gave them the P53 treatment, it was a little bit better, which I think statistically actually pretty close, depending on when you look at it, slightly better. And I think this kind of speaks to the overlap between these two, and also the fact that a lot of these mice will die from cancer at some point. But what was truly remarkable, and as far as I know, completely unprecedented, is that when we combined the two, they got both a treatment that killed P53 and one that killed P16. And as it stands now, there's one dead mouse out of the group. And I think this is completely unprecedented, partly because there aren't other tools that are really well suited to this. The transgenics could pick off one, but not both. And nobody has a vector that's even remotely close to the lipid nanoparticle we're using to deliver these. So, I mean, if you think about it, you need to have a treatment that you can administer multiple times at a high enough dose to hit a significant proportion of the cells in the body. And that pretty much rules out viruses, because you could only inject them once without eliciting a very severe immune reaction. You effectively vaccinate yourself against it. And the cost would be very prohibitive. You'd be looking at treatments that cost about $1 million a piece. But so this, like I said, this study is ongoing, and we're going to run it until the last mouse is standing. But we're super excited about it and super excited to share the data.
So that was a great update on the efforts of Oisin Biotechnologies. And there's no denying that senescent cell elimination is one of the hottest topics in regenerative medicine right now. Companies developing their own senolytic approach are popping up everywhere, and more funds are being provided. Juvenescense Limited, who recently completed a $50 million Series A financing round for life extension projects, has just closed a $10 million deal with the anti-aging drug discovery company Antoxerene. We spoke with Kelsey Moody, the CEO of Antoxerene, to find out more about this partnership.
Obviously, everyone is thrilled about our new partnership with Juvenescence, and we feel this deal is a strong vote of confidence in the quality of our work from a sophisticated partner. I think most everyone working in the life extension space believes that the first generation products will probably be senolytic drugs. We're really appreciative of pioneering work by others like Unity, who are de-risking the regulatory path for the entire field of senescence. Through our partnership with Juvenescence, we believe we can begin to deliver on the promise of this field. I do wish I could go into some detail about our research pipeline, but suffice to say that Antoxerene, and really Ichor's entire portfolio more broadly, is beginning to reach maturity. The next few years are going to be very exciting for us.
One of the organizations that are actively involved in research into senescent cells is the Methuselah Foundation. On their recently revamped website, they shared the touching story of the fictional Mr. Howie, a 90-year-old man living in a nursing home. The story, based on the real-life work experiences of a geriatric nurse, describes in detail the challenges and pain that patients like Mr. Howie experience on a daily basis. Some recently published opinion pieces contend that focusing on aging-related diseases should not be a priority, but this story could change some minds. While Mr. Howie may be fictional, his story is everyday life for many real people, and unless we focus on the science of aging, one day his story could be yours. We recently spoke with the story's author, Methuselah Foundation Research Fellow Danielle Ruiz, to find out more about her foundation's work.
The Methuselah Foundation is a non-profit organization that was co-founded in 2003 by David Goebel and Aubrey de Grey. Our mission here at Methuselah is to make 90 the new 50 by 2030. And we're doing that by supporting tissue engineering, regenerative medicine, and other novel approaches to extending the healthy human lifespan. Our approach is multi-pronged because the problem is multi-dimensional. We chose to have the very specific mission of making 90 the new 50 by 2030 because it keeps us committed to something we like to call return on mission. Return on mission is based on the return on investment concept of the financial world, except this deals with how we're achieving our mission. To us, return on mission is based on how many lives we've saved and how much suffering has been prevented. We recognize the painful fact that aging causes 100,000 deaths every day and creates immeasurable suffering for millions on a daily basis. Now, we could have chosen a vague or general goal like improving health or extending the lifespan, but we chose this very specific goal and deadline. The downside to us not choosing a grand statement with no deadline is that we know we are going to be judged come 2030, but we like that. We never want to become the type of charity that exists just for the sake of existing. We incubate and invest in early-stage life science companies. We fund scientific research. We also provide fiscal sponsorship to aligned projects. And we sponsor inducement prizes where teams compete to come up with scientific breakthroughs.
If you'd like to read the story of Mr. Howie, we've linked to it at lifespan.io/roundup. In other big news, the Biogerontology Research Foundation, in collaboration with the International Longevity Alliance, has submitted a proposal to the World Health Organization to include aging as a disease in the International Classification of Diseases, or ICD. Although aging is not yet classified as a disease, this proposal led to the WHO to introduce the new aging-related extension code via the ICD-11. The introduction of this new extension code shows that the WHO is receptive to the idea of aging as a dangerous but treatable condition. Dr. Alex Zhavoronkov from In Silico Medicine told us more.
In 2015, we formed a task force to look at the various ways to classify aging as a disease in the context of ICD-11, published several peer-reviewed papers, and the recognition of a condition or a chronic process as a disease is an important milestone for the pharmaceutical industry, the academic community, healthcare and insurance companies, as well as the individuals. This presence of a condition in the disease nomenclature impacts the way it is treated, researched and reimbursed. It highlights the condition. So right now we're witnessing the revolution in longevity biotechnology, and having a disease code in ICD is likely to provide at least some support for these pioneers.
For an example of what this new aging-related extension code could mean in practice, we spoke with Daria Kalterina of the International Longevity Alliance.
This code can be added to any of the huge list of diseases which are already in the system, which technically creates a large number of new aging-related diseases. For example, instead of immune deficiency, we get aging-related immune deficiency. This describes a new condition with specific aging-related underlying mechanisms such as naive T-cell depletion. As a result, new therapies against aging-related immune deficiency can be developed, registered, and prescribed by the doctors.
July also featured some great content on our website. We have new interviews with Dr. Aubrey de Grey and Dr. Peter de Keyser, an article from Elena Milova discussing her experiences at Open Longevity's recent anti-aging nutrition course, Steve Hill breaking down some important new discoveries involving telomeres, and resources to clear up the rampant confusion regarding overpopulation. We also have a new episode of our Journal Club video series, which explores recent concerns surrounding the gene editing tool CRISPR-Cas9. You can find all of these articles and more at lifespan.io/roundup. That's it for our first episode of the Rejuvenation Roundup Podcast. Since this is our first episode, we need you to help us grow, and it's very simple. All you need to do is subscribe to this podcast, leave a review, and post about it on social media. This will increase our reach and introduce more people to the importance of life extension science. Still want more? You can get additional deep dives into science, technology, and futurism on my Future Grind podcast. Recent episodes have focused on robotic avatars and virtual worlds. You can find these episodes and many more, including one on the science of life extension, at futuregrind.org. Once again, I'm your host, Ryan O'Shea, and on behalf of the team at Leaf, we wanted to thank you for joining us. We hope to see you next month on the Rejuvenation Roundup Podcast.